Good morning and good afternoon to call participants in Europe. Welcome to today's Chardan leadership call featuring LogicBio Therapeutics, a company focused on developing therapies for pediatric genetic diseases, leveraging the GeneRide nuclease-free editing platform and the Savvy capsid platform. And a special thank you to our featured speaker, Fred Chereau, Chief Executive Officer of LogicBio. I'm Gula Lifshitz, a biotech analyst at Chardan, and as call participants are aware, our focus at Chardan is on identifying companies that can generate exceptionally high long-term investment returns by creating and sharing in value for society. During this call, we will not be discussing Chardan research. Any discussions about Chardan research should be coordinated between our participants and their respective salesperson at Chardan. The recording of this call will be made available to clients after the call, and our compliance team has asked me to read this statement. By participating in this call, our speakers attest that they have made Chardan aware of any potential conflicts, and they will not discuss any material, non-public, or confidential information that they're aware of that may breach their legal, regulatory, or fiduciary responsibility to any parties. In terms of format, this is a 45-minute to 1 hour long call, and participants will be given the opportunity to ask questions. To ask any questions, email them to me at julipshitz@chardan.com or enter them into the Q&A Zoom window. So, Fred, thanks again for joining us today. And to start off, would you provide some high-level introductory comments on LogicBio as a company and its history for those that might be new to the story? Fred, you might be on mute.
Thank you. Thank you, Gula, for inviting us today, and thank you to the Chardan team for putting this call together. And also thank you for the audience for their interest today. So LogicBio is a clinical stage genetic medicine company pioneering gene editing and gene delivery platform. And we are intending to address rare and serious diseases from infancy through adulthood. So we are our gene editing platform that we call GeneRide is a new approach for precise gene insertion. We harness a naturally occurring process called homologous recombination. We believe that GeneRide is uniquely positioned to deliver therapeutic levels of protein expression through a nuclease-free gene editing technology with a one-time precise viral vector delivery of the therapeutic transgene and leveraging an endogenous promoter. And I'll tell you more details about that in a minute. The lead program coming from our platform is called LB001. It's a clinical stage single administration therapy for the treatment of MMA in pediatric patients as young as 6 months old. We recently announced that we have dosed our first patient. In addition to MMA, we are evaluating GeneRide in multiple other diseases, both in-house here in Cambridge and Lexington now, and through a partnership with companies like Takeda and Daiichi Sankyo. On the gene delivery side, we have also a next generation AAV capsid platform that we call Savvy. So Savvy is designed to optimize gene delivery for treatment in a broad range of indication and tissues, and that we can use for both gene editing and what we call canonical gene therapy. We believe the AAV capsid coming out of the Savvy platform have the potential to provide opportunities to superpower GeneRide, but potentially also address some indication in the liver based on our experience, and also with others that could not get a potency high enough to get in the needed expression. So we are developing this highly potent vector for the use in our internal programs, but also potentially for business development collaborations.
Great. And so before we dive into the programs in more detail, can you briefly take us through the key catalysts that are anticipated for the remainder of the year and into 2022?
Sure. So 2021 has been a busy year already. And on the clinical standpoint, obviously, as I said, we've announced recently our first patient in the Sunrise trial. And here we remain on track to provide updates on enrollment, dose escalation, and age de-escalation in late 2021. And the plan is to provide some interim clinical data by the end of the year, by the end of 2021. In addition, we are also planning to announce the next development candidate, in-house development candidate, later this year.
Great. So we'll be looking forward to those updates. So we've also heard a lot about gene editing during the past few weeks. Can you describe your unique approach to gene editing with GeneRide and how it may be distinct from others in the space?
Yes. So as many others, we were excited to see the latest news on in vivo gene editing disclosed by Intelia. And while our technology is quite different, we believe we have also a significant role to play in this space. So as I alluded to, our main differences compared to CRISPR-based technologies are the three following. The first thing, as very few of them, we are in vivo. And to date, Intelia, that's what Intelia disclosed recently. The main difference is we are nuclease-free. We don't use these molecular scissors to foster the integration. And also we are doing knock-in, which means we insert the gene in a specific locus of the genome, while, as Intelia described, what we have done is knockout. So one of the differences. We believe these characteristics are what allowed us to go into patients as young as 6 months old. And as GeneRide offers several key potential advantages over gene editing and gene therapy technologies that relies on exogenous promoters and nucleases. We also believe that by using or harnessing homologous recombination, GeneRide will allow us to precisely insert a therapeutic gene or transgenes into targeted genomic location without the need to use these molecular scissors I was talking about, the nucleases. It allows us also to drive high level of tissue-specific gene expression without the possible detrimental issues that has been associated with the use of exogenous promoters in the case of off-target integration. So talking about precision, our homology arms are made of hundreds of nucleotides, between 500 to 1500 on each side, depending on the gene we are using. In contrast to RNA guide sequences used in CRISPR-Cas9, as you may know, are made of only 20 nucleotides long. And we believe this increased length improve both the precision and the site integration. In traditional gene therapy, and I'm sidetracking maybe a little bit, but the corrective gene, as you know, do not integrate in the patient's chromosome and remain floating inside the nucleus. And the corrective gene is not carried out when the cells are dividing, so the therapeutic effect is diluted out over time. As we are harnessing the cell natural DNA repair process, the homologous recombination, and we precisely insert the correct version of the missing gene in the cell genome. The gene that persists as part of the patient's DNA as the cells are dividing, give us the potential to lead to a more durable effect, and especially when you target pediatric patients, when the liver is either growing or dividing due to the disease. And lastly, another word on our platform, which is, as I said, we are promoterless, and we don't carry an exogenous promoter. We take advantage of an endogenous promoter, and we believe, as I said, that in case of off-target integration, it would be reducing the risk of cancer, as which could be the case in some more traditional gene therapy approach. So when you put all these things together, we think that GeneRide is well positioned to treat genetic diseases, but specifically in pediatric patients and when this disease have an early onset.
Great. That's a great overview. And so Maybe let's start on talking about methylmalonic acidemia, and then we'll talk about some of the capsid work in more detail afterwards. So as you mentioned, in the beginning of June, you announced the dosing of the first patients in the MMA study. Can you give us a brief overview of the disease and the therapeutic approach there, and how it might be distinct from what others have been doing in the indication?
Yes. So yes, as you say, I mean, we were thrilled to announce that our first patient has been dosed. I think it's a significant milestone for a young company and significant milestone in the development of LB001 for pediatric patients with MMA. But also, I think a significant milestone for the field in general, as we believe that dosing this first patient with an in vivo gene editing technology would be a good proof of concept for our platform to deliver systematically to pediatric patients. So I think we are the first ever in vivo gene editing delivering systematically in a child. And it's also, I mean, it's an important milestone for the company, but for the MMA community and the overall genetic medicine field. So talking about MMA, there are no currently there are no approved treatment options for targeting the underlying cause of the MMA disease and treating the enzymatic defect in MMA. And because of that, we know that it's critical to prevent the progressive irreversible tissue damage that leads to long-term complication. The only tool or management that the physician can offer today to patient is actually just a restrictive low protein and high calorie diet, and which is very often administered through a feeding tube. And however, despite this pretty difficult to follow diet, metabolic crisis and long-term complications still occur, and these patients have a very high rate of hospitalization. So ultimately, liver transplantation is another option, and it's performed in severely affected patients. However, not all patients experience normalization of liver and renal function or improvement of the neurodevelopmental status. And as you know, the transplantation carries its own risk and carries also a lifetime need for immunosuppression. So we expect that LB001 will improve metabolic stability in this disease, which is also a key benefit of the liver transplant, but without being associated with the morbidity and mortality and without the wait for a suitable donor.
I see. And so LogicBio also announced results from a retrospective study earlier this year in MMA. So can you talk about what you learned from that study in terms of the natural history, disease markers, and the metrics of unmet need, and how that might guide your thinking on the LB001 program?
Yes, so the study results were recently highlighted in a poster presentation at the American College of Medical Genetics and Genomics annual clinical genetic meetings in April 2021. It's a retrospective non-interventional natural history study where we collected the data via chart review, mainly in MMA patients. The results showed a significant and sustained decrease of methylmalonic acid levels, which is the toxic metabolite, post-transplantation compared to baseline level. While the level did not approach normalization, there was no apparent reduction an apparent reduction in variability and a potential indication of improved metabolic stability. So that was good to see that the liver transplantation has an effect on this patient. The results indicate that there is no significant difference in MMA or transplant-related emergency room visits and stay or hospitalization among the patients pre and post-transplantation up to 3 years. However, we believe that the interpretation of the data and the healthcare use was limited as the data of the for 2 and 3 years post-transplant were available only in 1 or 2 patients. So it seems that transplantation has an impact on ER visit, hospital stays, and overall hospitalization. But I think we didn't have enough data to be more affirmative on this specific endpoint.
Mm-hmm. I see. And does that impact the thinking or the design of how you're planning the LB001 program?
Well, the LB001 program was already on the go when we were doing this study, so it has no impact on this program. But that's definitively interesting findings that we will keep with us for the future of the whole program. As you may know, we are also running a prospective natural history study, which I think should be more informative, but we'll tell you more about that in the future.
Great. And so in terms of NNA as a biomarker, what kind of NNA or other biomarker modulations would be considered clinically meaningful in this patient population?
So as you know, I mean, that's the first time there is a clinical trial which is testing a drug addressing the underlying cause of the disease. So we still have a lot to learn and about the biomarker modulation and what would be considered clinically meaningful. But first, I would say the discussion with the regulatory agencies will determine that and will help us will guide us in how we're gonna look at how these different biomarker, MMA and others, are evolving after treatment. For now, after the diet, the next the standard of care in liver transplantation, we know that after the biomarker like MMA and others are significantly decreasing, and we believe that LB001 has the potential to, as I said before, to improve the metabolic stability, which is a key benefit, but also to decrease MMA levels and other important biomarker in this liver disease. So sorry for not being more precise, but I think we need to have more experience on the clinical stage to be more definitive on this question.
Mm-hmm. Got it. And so with regard to the clinical trial, can you walk us through the clinical trial design of the Sunrise study with respect to the enrollment criteria and the dosing cohorts that are ongoing and planned?
Yes. So Sunrise is a first-in-man, 50 weeks, multi-center, open-label phase 1/2 study. It's designed to assess the safety and the tolerability of LB001 in pediatric patients with severe MMA associated with the MMUT deficiency. As you may know, some MMA patients have other mutations, but the largest number of MMA patients are carrying this mutation in the MMUT gene. So in addition to dosing the first patients, we have now opened several sites and identified enough additional patients to enroll the 8 patients that we plan to enroll in Sunrise. Obviously, it's subject to screening clearance. The trial is initially enrolling pediatric patients between 3 years old and 12 years old, with the possibility to include patients as young as 6 months old after meeting certain safety parameter and detection of a specific PD biomarker that we called albumin 2A, which is a byproduct of our technology. So LB001 will be tested at 2 doses. The first dose is 510E13 Vg per kilo, and the highest dose will be 110E14 Vg per kilo. So after initially started at the lower dose in these 3 to 12 years old patient group, we will do age de-escalation and dose escalation. So the decision to escalate the dose will be determined by safety solely, and whereas the age the decision for age de-escalation will be based on both safety and detection of albumin 2A, this PD biomarker I was talking about. The FDA required that there is a 6-week staggering interval between the dosing of each patient. And with the exception at the age de-escalation and dose escalation that we could that can occur simultaneously. What I mean here is after the first 2 patients in the 3 to 12 years old group at the low dose, we could in parallel enroll in the same age group at a higher dose and at the same dose in a younger patient population, 6 months to 2 years old. 2 patients in each group, here again, with 6-week staggering. And then at the last group will be of 2 patients in the youngest group at the highest dose if it's needed. So I hope I was clear. It's a little bit of a complex design, but it's a very interesting design to test the dose in different ages, and especially to allow us to go to younger patients as quickly as possible, because as you know, that's what physicians are requiring in some point. Everybody knows in the field of MMA that the earliest you can intervene in this patient, the better it is for their future.
Right, that makes sense. And so you mentioned safety and the biomarker activity. On safety, safety with systemic AAV dosing has continued to be a topic of interest and discussion within the genetic medicine space. Can you talk about what the study is doing to maximize safety with the administration regimen?
Yeah. So first, I mean, we had a robust preclinical program in different animal species, and it's by combining all this data and also with extensive discussion with the FDA that we came up with the dosage scheme. You know? And you know that in genetic medicine, gene therapy or gene editing, since to date you cannot, when you treat with AAVs, you cannot go back to this patient. You want to make sure that the first dose you will use will be an efficacious dose. So we spend a lot of time here triangulating the data to make sure we were in a potentially an efficacious dose. I will also note that in our overall preclinical program, we did not see any safety signal, you know, and that's also very encouraging. But the patient which will participate to the trial will go first to a pre-dosing running period, and then they will be administered a prophylactic steroid regimen that begins prior to the treatment, as it is fairly standard in many other programs testing this type of therapies. So nothing, I mean, very different, and we are using like prophylactic steroids, as I was saying.
I see. And then in terms of the other endpoints of the study, can you walk us through the endpoints with regard to the type of biomarkers that will be measured, that have been disclosed? And additionally, can you talk a little bit more about the ALB2A biomarker and what that means for the interpretation of the data based on what you've seen in the preclinical settings?
Sure. So about the first endpoint, I mean, is a safety endpoint, obviously, but I guess everybody is interested in also seeing how efficacious this treatment could be. And maybe a little bit of, I'd like to remind you a little bit about this PD biomarker that we call albumin 2A. It is a protein which is produced when an edited hepatocyte is expressing the MUT protein, and which will stay intracellular and will traffic through the cells and the mitochondria. Sorry, I was looking for the word. But at the same time, because we are doing what is called a bicystronic protein expression, which means that with only one ribosomal read, we can express two distinct proteins at a 1-to-1 ratio. We have this albumin on which we have taken a ride on to take advantage of this potent promoter, which is tagged by a small peptide called 2A. And this albumin goes to the circulation, and we can measure this albumin 2A among the normal albumin in the circulation. The studies have shown that it can be very interesting. And in preclinical mice studies, the detection of albumin 2A not only indicated the integration, that the drug has been integrated in the hepatocyte, but also correlated with therapeutic benefits from LB001, such as increased body weight and also declining of circulating methylmalonic acid, which is once again the toxic metabolite. So, as I said, albumin 2A is measured by taking blood samples, and therefore we are avoiding invasive liver biopsies that you want absolutely to avoid in this type of patients. So the Sunrise trial tests the patient blood sample for albumin 2A at very specific intervals. And we believe that based on the preclinical mice studies, the detection of albumin 2A followed by the increased level of albumin 2A will be a good indication of a successful integration of the corrected transgene, but also will have the potential to correlate with therapeutic benefits, as I said just before, such as body weight and the decline of circulating metabolic acid levels. In terms of timing, it's difficult to know, and that's the reason why we do the trial. But in mice, the detection of albumin 2A is quick, and in a matter of days to a couple of weeks max. The timing in human, as I said, is unknown, and we hope to be able to see that quickly in the Sunrise trial.
Mm-hmm. Got it. Great. And in terms of MMA as an indication, we understand that that can be an indication that is challenging to enroll. You've talked about pre-identifying some patients for the MMA study, or the Sunrise trial, I should say. Can you talk about how efforts to pre-identify these patients have gone and your engagement with the patient community and regulators and clinicians in the space?
Yes, so from the beginning of the program, we have been very close to the physicians, the key opinion leaders, as well as the patient advocacy and patient families too. And we think that is helping us, has helped us to pre-identify many of these patients, and will continue to help us to enroll efficiently in the trial. I think what was challenging, it was more during the COVID outbreak to open the investigational sites. It took longer than what we were expecting. And now we continue to work on activating additional sites, and we believe that after the first patient that we have announced recently, and also respecting the 6-week staggering in between each patient with the little caveat I was describing before, we think we are now on a good track to manage the trial efficiently. So I think that the key for us was very much to develop this strong relationship with the patient community and the physicians, and we are lucky to be able to work with among the best physicians in the US in this disease.
Great. And in regard to the expected catalysts that you talked about earlier with the operational update and the initial data readout, can you give us a little bit more color on what we should be expecting from those updates?
Yeah, so what we call the operational update is basically when we move from one group, one subcohort to another. As I said, initially we are enrolling a patient as young as 3 years old, and when we will detect safety, when we will confirm safety, we'll be able to go to a higher dose. When we will confirm safety and detect this PD biomarker, albumin 2A, we will be able to go to a younger group. So that's something we will share with you. This move to another subcohort. If you refer to the slide on our corporate deck, which is online, you will see that there is a group on the right, a group on the left, and we very often internally refer to that. So on the right, same age group but higher dose. On the left, same dose as the initial subgroup, but younger age. So I think when moving to the right and increasing the dose will be a good signal of safety, but more interesting, moving to the left would confirm that we have detected albumin 2A, which is once again a proof of integration, so that drugs is integrating into the genome, and expression of the missing protein. So we think that's going to be very important information to share in the coming month. Now, I think you were also referring about the end of the year, where we said that we will disclose more safety and a larger pool of safety and efficacy interim data. And it's difficult to say today how large this pool will be. Because it's obviously we need to confirm this goals we have for enrollment, but we will provide additional data at an appropriate time.
Mm-hmm. In terms of the kind of data that we should be expecting, is that along the lines of safety and potentially biomarker data, or how should we be thinking about the type of data that we should expect for that initial readout?
Yes, obviously safety is important, but as if you refer to the clinicaltrial.gov, our page on the clinicaltrial.gov, you will see that the secondary outcome measures are things such as the changes in serum methylmalonic acid and methyl citrate. We are also looking at FGF21. We are looking at proprietary net oxidation. We are looking at albumin 2A, obviously, and overall survival and other clinically relevant endpoints. So we'll try to disclose as many data as we have, assuming they are relevant to share this at this time point, end of the year, before the end of the year.
Mm-hmm. Great. So yeah, absolutely looking forward to that. And looking longer term ahead, based on your interactions with regulators, do you have a sense of what kind of endpoints would be most important for approval in this indication?
So, being the first company trying to tackle this disease, it's a new ground for us. It's a new ground for FDA too, and or any other regulatory agency. So we don't have yet a full answer. What we have is regulatory designations such as Fast Track and Rare Pediatric Disease Designation and others, which will allow us to have a good dialogue with the regulatory authorities as we will be accumulating knowledge on how this drug is affecting this disease and what would be relevant to continue to survey and what could be kind of endpoint for the approval of the indication. So give us a little bit of time, and here again, we'll share as much as we can.
Got it. And then maybe I'll get a similar answer, but in terms of potential regulatory paths, are there potential scenarios that would be appropriate for a regulatory path in this type of indication and with this type of therapy that you're keeping in mind?
There are many scenarios we are keeping in mind for under regulatory path here, but I think here again we will provide you with more details as we will have more certainty about the plans we want and we can follow.
Great. And then coming back to the gene editing space more broadly, as you mentioned, the gene editing space had positive data from the first systemic CRISPR therapy. So do you see any read across for LogicBio from either a scientific, regulatory, or other standpoint?
Well, sure. I mean, I think the first one is it's great to see that this first, to my knowledge, at the first time an in vivo gene editing technology is demonstrating some positive data in a clinical trial. You know, it's early data, it's a limited number of patients, so it's important to remain very prudent. But to date, I don't think anybody has done it before. As I thought, I mean, for us, the read across is in vivo gene editing seems to work. And what we will intend to prove is that it can also work without nucleases, and if we do so, and without, I mean, by doing some knock-in, which has not been proven to date, which means to insert the gene in a specific locus of the genome. As I said before, Intelia has done some knockout, which is already very interesting. But as we intend to prove that we can do in vivo gene editing, knock-in, and doing so, it would be opening the door for treating early onset diseases and treating the patient when it's, I mean, as early as possible before some non-reversible sequelae occur. And that's one of the reasons why we decided to pursue this program in methylmalonic acidemia, which, by the way, is one of the few disease on the newborn screening panel in the 50 states for which there is no therapeutic treatment.
I see, right. That makes sense. So maybe now let's shift gears and talk a little bit more about the Savvy capsid platform that you introduced in the beginning. You mentioned the platform, and LogicBio has presented some data at ASGCT on the first capsid coming out of that platform earlier this year. Can you explain the platform for us and discuss how it's designed to address some of the key challenges in the AAV-based gene therapy and gene editing space, such as safety and potency?
At first, I would say that, I mean, we gave a name recently and we call it Savvy, as you mentioned, but from the beginning, the inception of the company, we've been developing these two platforms, the gene editing cassette, as well as the gene delivery, innovative gene delivery system. And the first one we have used is coming from our scientific founders out of the lab of Marquet at Stanford University, and is called LK03, and is in the clinic now with Spark, but also with us, because that's the capsid we are using in our LB001 trial. So as you know, I mean, natural and engineered serotype capsids are very often screened and selected in mice. You know, and now people are moving to monkeys, but what we have learned over the past decade is that it's not because a capsid works very well to target a specific tissue in a mice that it's going to be the same in human. And actually, we've seen that the efficiency of these capsids is decreasing when we move to mice to NHP and from NHP to humans. So the Savvy platform, however, use an innovative way to first develop or engineer this very diverse synthetic capsid libraries, and then to screen them in what we believe to be clinically relevant animal models, such as a chimeric mice model we have been using for a long time, and other models or other tools we are using now, because we have enriched a lot the work which has been done initially at Stanford in this collaboration we are having with CMRI, where Leszek Lizowski, with the L of LKO3, is a professor now, sorry. We have the third point we have added to the platform is product development, and we have now seen outstanding production yield with a proprietary process we have developed at LogicBio. And when we compare to published data in AAV yields, we have very, very interesting data. As an example, and you mentioned this new capsid SL65. SL65 has shown first an enhanced functional transduction in the humanized mice, and we think it's a very good predictor of what's going to happen in humans. When you compare to LKO3, which is already we believe it's the best capsid for the liver in the clinic to date, but SL65 is 2 to 3 folds better than LKO3, and importantly, 10-fold better than AAV8, which has been the most used capsid maybe in liver gene therapy. We have also in NHPs, we have shown a higher factor 9 expression level compared to LK03 or AAV8 once again. And we have recently demonstrated some very interesting data at ASGCT, where we've shown that SL65 has a higher liver specificity in NHP, but also a uniform distribution throughout the liver, which could be, we believe, important in certain diseases. And that's also something we hope to share more in details as the time will pass. But, and lastly, as I was mentioning before, I mean, the PD, the capsid we have developed, in addition to the technologies we have improved here at LogicBio, have demonstrated a very high manufacturability. And so we think that as SL65 is approaching the clinic, it could be a very significant capsid for this genetic medicine, AAV genetic medicine targeting the liver. Maybe the last point I would say, and it's linked with manufacturability, it's about the cost of goods, or moving to larger indication. So when you combine a higher transduction efficiency that I was mentioning, and a better immunological profile, I didn't talk about that, but it's also something we are paying attention to when we select these capsids. We looked at, I mean, the overall profile. Would it be a neutralizing, like pre-existing neutralizing antibodies, or other factors you want to look at for capsids? But combined with this outstanding production yield, we think these three levers are very important to reduce the cost of goods and potentially move to a larger indication. So we believe there is a real opportunity to deliver some interesting value for companies, but more importantly for patients, and a larger number of patients here.
Great. And so right now it sounds like the focus is on the liver. Can this approach be extended to other tissues?
Yes. So we focused on the liver because I mean, at the K-Lab, people had a lot of experience on the liver, but also we noticed that there are very few programs targeting the liver which can address early onset genetic diseases. And then we thought we could be, and we are uniquely positioned to target those very young patients, which still have a pretty, what I would call, active liver, either growing or regenerating due to the tension of the disease. We have already demonstrated and published and presented actually some data in other GeneRide data in other tissues, and we have shown interesting data in fibroblasts and keratinocytes, mainly to demonstrate that we could take a ride behind another gene than the albumin. And the albumin is used in the liver as you know, it's one of the most potent, most highly expressed gene in the liver. Now, recently we announced an extension of the collaboration with CMRI. It's a two-year extension, and we also announced that we are doing research of new capsids for 2 additional tissues. So because GeneRide is a pretty modular system, when you have the right capsid to target the right tissue, we know how to tweak or change this homology arm to target a specific locus in the new cell you want to go after, and insert in the middle the gene you want to, the transgene you want to express, coupled with this small 2A peptide, which is once again a very interesting feature, which helps us to track how the treatment, or at least the technology, is effective in these patients.
I see.
So more tissue to come, yes.
I see, got it. And so earlier you mentioned some collaborations that LogicBio has with Takeda and Daiichi. Can you talk a little bit about those collaborations and LogicBio's role and what we should be looking out for from those collaborations?
Yes, so we announced the Takeda collaboration a little bit more than a year ago. At the time, we were still a preclinical company, and the IND was not even filed. So we are evaluating another product candidate beyond MMA, which is called LB301 for Crigler-Najjar syndrome. In addition to the GeneRide collaboration, we have another GeneRide collaboration with Daiichi Sankyo for 2 other indications, which has not been disclosed yet. Both of them are research collaboration, and our partner are providing funding for the research program under the collaboration agreement. And we are doing a big, if not a large part of the work here in-house, and we will have, and then we will have an exclusive option to negotiate an exclusive worldwide license for the corresponding program. In addition to these 2 programs, which are GeneRide related, we have announced a collaboration with Cambridge Pharmaceutical, a Chinese company, under which we granted Cambridge a worldwide license for SL65, the capsid I was talking about, and the first capsid we produced out of the study platform. They have a license for the development and the commercialization of gene therapy candidates for the treatment of Fabry and Pompe disease. We also granted Cambridge an option to obtain a license, the same type of license for 2 additional indications. And finally, they also took an option for a development and commercialization license for LB001, the MMA program in Greater China. So that's the 3 collaborations we have to date.
I see. And so, at a high level, thinking about the capsid development platform, how does that technology platform feed into the business development efforts as we think about LogicBio moving forward? And yeah, with these deals in place now, how are you thinking about collaborations more broadly going forward?
Yeah, that's a very good question. And we have already proven through this collaboration with Cambridge Pharmaceutical that some people are interested in using this capsid and taking them to the clinic. So it's kind of a classic capsid out licensing. That's opportunities we want to continue to tap on, and we continue to generate data to demonstrate the superiority, the potential superiority of SL65 in other models and other construct. So definitively a pretty rich area of business development opportunities here. As you know, many companies most of the gene therapy and editing companies using AAVs are always looking for the most potent capsid for their program, and that things which that choices which happens very early on in their development. So there is a lot of active program ongoing, and we hope to be considered for this program. Now, we can, thanks to the collaboration we have with GeneRide, we can go beyond LSL65 in two ways. We continue to look for better capsids, so maybe there will be an SL66 or whatever we will call it, which will present more interesting features. But also, as I said, we have started programming two other tissues, and tissues where there is definitively a lack of good capsids, synthetic capsids, but capsids which once again would be selected in relevant models. And then there are many other therapeutic areas or tissue where selecting a capsid for its ability to transduce some mice cells in the specific tissue is definitively not the right thing to do. And we intend to bring our know-how and experience here to accelerate the development of a new class of capsid in other tissues.
Great. And can you talk about LogicBio's current approach towards manufacturing and how you might see this evolving as the Savvy platform matures and as the internal programs mature as well?
So to date, and for LB001, we have been working with CDMOs. But as you may remember, from the beginning of the company, we have heavily invested in process development capabilities, and that's thanks to that. I believe that we have been able to demonstrate outstanding production yields. So we intend to continue to develop this in-house PD capabilities to larger volumes. I mean, larger vessels, you know. It's part. It's taking a decent amount of our resources today. As the programs in-house and partner program will continue to evolve, we'll see how we continue to climb the food chain of manufacturing. That's something we will discuss maybe in the future. But we think this early work, which has been done from the initial selection, design and selection of these capsids, is very important to confer some low cost of goods and again to being able to provide our drugs to the larger number of patients, would it be in larger indication, but also throughout the world where the economical conditions are not exactly the same as in the US. So we continue to grow on this side.
Right, got it. And then with respect to cash runway, what is LogicBio's current cash runway, and how is the company thinking about deploying investment between internal pipeline versus platform advancement, and is that something that you also see evolving going forward?
Yes, so on the cash runway, so as of March 31, 2021, our cash equivalent were 63.9 million, which did not account for the $10 million upfront from the collaboration with Cambridge. We don't disclose any other information regarding our cash runway, but you can look at the historical data, and it will give you a good idea of where we can go. And your question was about the balance between a pipeline and platform, or investment in pipeline and platform. It's definitively evolving. We expect to continue to develop both internal programs as well as I said before, evaluate business development opportunities. So it's difficult to say today how it will evolve, but that's something we'll continue to monitor closely.
Great. And so in the last few minutes, I wanted to ask, based on your experiences and discussions with investors over the past several months, what do you think investors are currently most missing from the LogicBio story?
That's a great question, Gula. If I may, I would say a lot, but in 4 points, maybe I'd say the first is we are one of the 2 companies with in vivo gene editing program in the clinic. The other one being Intelia, as you know. Now, to our knowledge, we are the only one having ever dosed a pediatric patient systematically in vivo. So that's the second point. The third point is also, to our knowledge, we are the only company to date doing in the clinic in vivo gene integration, this knock-in I was referring to, through gene editing, which is the only way to do so. And lastly, we were just talking about the platform, the Savvy platform. I think we have a very strong AAV capsid development platform, which started, as I mentioned, with LK03, which is now proven in the clinic to be efficacious in the clinic through Spark, and hopefully soon through our clinical trial. And we believe that people do not totally realize the overall value of this capsids platform, which has evolved dramatically over time and is presenting great data in terms of efficacy and manufacturing yield.
Fantastic. So I think that brings us to the end of this leadership call. And again, I'd like to thank you, Fred, for your participation and the great discussion.
Thank you, Gula, and thank you to the Chardan team. Looking forward to meeting you in person.